Assalamu alaikum, wonderful students. Today we will discuss about plant hormones. What are the subtopics that we are going to discuss here are definition, characteristics, classification, and plant hormones actions. If we'll talk about the definition, there are various organic compounds other than nutrients produced by plants that control or regulate germination, growth, metabolism, or other physiological activities, also called phytochromes. Phyto means plants, hormones, and recently called growth bioregulators. Plant hormones and growth. It is the diagram that depicts the hormonal interactions that influence plant growth and development. Okay, definition: plant hormones, which are active in very low concentrations, are produced in certain parts of the plants and are usually transported to other while they are elicit specific biochemical, physiological, and morphological responses. They are also active in tissue where they are produced. Okay, definitions: each plant hormone evokes many different responses. Also, the effect of different hormones overlap and may be stimulatory or inhibitory. If it generates a response, it will be stimulatory. If the response is positive, if the response is negative, then it will be inhibitory. The commonly recognized classes of plant hormones are the Auxins, Gibberellins, Cytokinins, Abscisic acid, and ethylene. Clear? I'll repeat: Auxins, Gibberellins, Cytokinins, Abscisic acid, and ethylene. Some evidence suggests that the flower initiation is controlled by a hypothetical hormone called florescence. Gens means producing, flori means flower. ठीक है? That means flower initiation by florescence. But these substances remains to be identified. A number of natural or synthetic substances such as bracin, morphectin, and other growth regulators not considered to be hormones nevertheless influence plant growth and development. Okay. Each hormone performs its specific function. However, nearly all the measurable responses of plant to heredity or environment are controlled by interaction between two or more hormones. May occur at various levels. You can say the interactions may be the synthesis of new hormones, hormone receptors, and secondary messengers, ultimate hormonal action may be cooperative, antagonistic, or in balance. Plant hormones or plant growth regulators are internally secreted chemicals in plants that are used for regulating the plant's growth. Standard definition is signal molecules produced at specific location that occur in every low concentration in a very low concentration and cause altered processes in target cells at other locations. Characteristics: the concentration of hormones required for the plant response is very low, that is from 10 raised to power minus 6 to 10 raised to power minus 5 m. Comparing with the requirements of minerals and vitamins for plants, the synthesis of plant hormone is more diffused and not always localized. It means that most of the plant hormones that act are diffused from different sites. Very rare they are localized. If we'll talk about the classification, classes of plant hormones include promoters and inhibitors. Promoters are those that cause faster growth, and inhibitors are those that reduce the growth. Examples of promoters are auxins, cytokinins, gibberellins, and brassinosteroids. Inhibitors include ethylene, abscisic acid, and jasmonic acid. What do hormones control in plants? Actually, the roots and shoot growth, seed germination, leaf fall, disease resistance, fruit formation and ripening, flowering time, bud formation, anything related to plant growth is controlled by them. Plant hormone and their action are given in this table. Look at here. It is auxin. Example is indolystic acid. Structure is given. Uh, they are synthesized in embryo, meristems, buds, and young leaves. And function is to stimulate stem and root growth, promotes cell differentiation in tissue cell, culture, and in procambium regulates development of fruit and apical dominance cause phototropism and gravitropism. Okay, if we'll talk about the cytokinins, example is zeatin structure is shown in this diagram. They are synthesized in roots, promote root growth and differentiation, stimulates cell division and growth in tissue culture, stimulates germination and retards aging. Okay. If we'll talk about the gibberellins, example is G83, gibberellic acid 3. It is shown in the diagram. It is synthesized in meristems and young leaves and embryos. It promotes seed germination and bud growth, promotes stem elongation and leaf growth, stimulates flowering and fruit development. Okay. Uh, if we'll talk about the abscisic acid, its structure is given in this diagram. It is synthesized in leaves, stems, roots, and fruits. Major function is to inhibit growth, doses stomata during it closes stomata during water stress and promotes dormancy. Ethylene structure is given in this diagram. It is synthesized in ripening fruits and aging leaves and flowers. It promotes ripening of some fruits and thickening of stems and roots. Brasinosteroids example include brasinolite. Diagram is given here. It is present in seeds, fruits, shoots, leaves, and flower buds. Major function is auxin-like effects, inhibits root growth and retards leaf abscission, promotes xylem differentiation. Clear? Okay, here is the structure of acetic acids. Tryptophan structure is given here. Indolestic acid structure is given here. 2,4 or dichlorophenoxy acetic acid structure is given here. 1 naphthalene acetic acid structure is given here. If we'll talk about the auxins, auxin is general name for a group of hormones that are involved with growth responses. What are the growth responses that are produced by auxins? These are to elongate cell, stimulate cell division in callus. Callus are differentiated tissues. Actually, they are differentiated for attaining their destinated tissues. Not surprisingly, the term auxin is derived from the Greek word to increase the growth. This was the first group of plant hormones discovered. Discovery of auxin. Light is fallen on the tip, illuminated side of colipetile and shaded side of colipetile. Growth differences noted actually. Light is fallen and all the phenomena is discussed in this diagram. You better understand when you take it a closer look. While taking a closer look, you will understand that light falls on a coleoptile, that tip has been removed and the tip is covered by an opaque cap, uh, and tip is covered by a transparent cap, base covered by opaque shield, and the results that are given. Okay, these are the three experiments by which we see the discovery of auxins. Auxin is made in actively growing tissues, which include young leaves, fruits, and especially the shoot apex, and it is made in cytosols of the cell. Transport. It is basipetal or polar. Transport auxin is transported in a basipetal towards the base or base seeking direction. Auxin move from the shoot tip towards the root and from the root tips towards the shoot. Clear? Function cellular elongation. It induces and amplifies proton pumping. While amplifying proton pumping, it means it is enhancing the cellular elongation. Acidified cell walls have increased elasticity, which lead to cell elongation. Second function is cell differentiation. Auxins promote differentiation of vascular tissues. Vascular tissues include xylem and phloem, auxin and sugar vascular tissue, auxin and low sugar xylem, auxin and high sugar phloem, auxin and moderate level of sugar xylem and phloem both. Ethylene production, third, is the function is indolystic acid apparently stimulates the production of ethylene. Fourth function is inhibition of root growth. Inhibits root elongation and very low favors root elongation, and this is the concentration actually at which it inhibits or favors the root elongation. Fifth one is to stimulate root initiation. Lateral roots, adventitious roots. Roots always form at the basal end of cutting. Formation of roots. Sixth function is flowering. Although most plants don't initiate the production of flowers after Auxins treatment, pineapples and its relatives, known as Bromeliaceae, do. Once flowers are initiated in many species, indole acetic acid promotes the formation of female flowers. Okay. Seventh function is parthenocarpic fruit development. For parthenocarpy, pollination of the flowers of angiospermic initiates the formation of seeds. Parthenocarpy is actually the ripening of fruit without fertilization. As the seed matures, the releases auxin to the surrounding flower plants, which develop into the fruit that covers the seed. Some commercially growing deliberately initiates fruit development by applying auxin to the flower. Not only does this ensures that all the flowers will set fruit, maximize the likelihood that all the fruits will be ready for harvest at the same time. Okay, auxins actions. It action is apical dominance. It improves the growth at apical regions. Lateral branches growth are inhibited near the shoot apex but less so farther from the tip. Apical dominance is disrupted in some plants by removing the shoot tip, causing the plant to become bushy. Eighth character is apical dominance. Ninth character is tropic responses, such as gravitropism or phototropism. Gravitropism is the response towards the gravity, and phototropism is reverse response toward the light stimulus. If we'll talk about the phototropism, sunlight breaks down the auxins. Plant stems in direct sunlight will have the least amount of auxin. Area of the plant that is more shaded will have more auxin. More cell growth on shaded side, plant bends towards the light. It means that this is the example that is shown. Light directly over the plant, auxins are in equal quantity. Cell elongation is equal in all sides of the cell. Greater light on the light side of the right side of the plant, auxins quantity become greater on the left side. Auxins trigger cell elongation on the left side. Plant stretches to the right. Is that clear? Okay. Geotropism or gravitropism. The plant stem that was once upright is on its side. The auxins are settled on the bottom side of the stem. More auxins accumulate on the bottom side. More cell growth occurs on the bottom side. Plant bends upward. A growth response to gravity which causes root to grow downward and shoots to grow upward. Clear? Okay. Gravitropism action of a due to auxin is shown in the diagram. This is the gibberellins. Three of the more than 80 gibberellins. Gibberellic acid 3, gibberellic acid 13, and gibberellic acid 4 are given in this diagram. Gibberellins are plant hormones that promote growth, seed germination, and leaf expansion. They occur at low concentration in vegetative tissues but at higher concentrations in germinating seeds. They induce cell elongation and cell division, important for plant growth and development through flowering and or seed germination. If we'll talk about the gibberellins, the site is young leaves, roots, and developing seeds and fruits. Transport made in the tissue in which it is used. Transport occurs through xylem, phloem, or cell to cell. Phloem seems to be most important transport route. Transport is not polar as it is for auxin. Clear Gibberellins actions. It promotes cell elongation, stem elongation specifically. When applied to intact plants, gibberellic acid usually causes an increase, unlike auxin. It overcomes dwarfism. Dwarfism is actually the short type, okay? In mutants that have a mutation in the gibberellic acid synthesis pathway. If we'll talk about the dwarf, that means short, wild type, tall, and dwarf plus gibberellic acid also have tall results. Thus, gibberellic acid application stimulates elongation and acts on an intact plants. Second action is to overcome dormancy in stems or seeds. Actually, dormancy is the plant time period where it is resting, not in an active state. Gibberellins also have a fundamental role in breaking seed dormancy and stimulating germination. The endosperm of many seeds contain proteins and carbohydrates, um, a reserve upon which a developing embryo relies for energy and nutrition. These reserves must be mobilized and transported to the embryo. A range of hydrolytic and proteolytic. Hydro means water, lytic means breakdown, and proteo means proteins, and lytic means breakdown. Breakdown endosperm starches and proteins into smaller, more easily transported molecules such as sugars and ab- amino acids. These are the diagrams that show the mode of action of gibberellic acid to overcome dormancy in seeds. Third is involved in parthenocarpic fruit development. Fourth is can induce fruit enlargement. External application of gibberellins can also enlarge fruit size in grapes. Fifth is it promotes cell division and elongation. Sixth is that it induces flowering. Gibberellic acid stimulates bolting in long day plants and can substitute for long days or cold treatments that are necessary for flowers. Okay. Seventh is sex expressions. In plants with separate male and female flowers, gibberellic acid application can determine the sex. For example, in gibberellic acid treatment increases the proportion of male flowers. Clear? If you want male flowers, then you can spray the gibberellic acid on your plant variety. In maize, gibberellic acid treatment causes female flower development. Here are the cytokinins diagram: kinetin, adenine, zeatin, riboside are shown in the diagram. Cytokinins are hormones that stimulate cell division or cytokinesis. Kinesis means division of nucleus or cytoplasm. These hormones may also be involved in controlling leaf senescence and the growth of lateral branches. The most active naturally occurring cytokinin is zeatin. Cytokinins occur in the most plants, including mosses, ferns, conifers, algae, and diatoms. Site synthesized primarily in the meristematic regions of the root. Actually, these are the regions that divide and redivide. This is known in the part because roots can be cultured, growth in an artificial medium in a flask without adding cytokinin, but stem cells cannot. Cytokinins are also produced in developing embryos. Transport It transport occur via xylem. Zeatin ribocytes are the main transport forms converted to the free base or glycosides in the leaves. Some cytokinins also move in the phloem. If we'll talk about the cytokinins action, control morphogenesis. In plant tissue culture, cytokinin is required for the growth of a callus. Callus is an undifferentiated tumor-like mass of cell. If we'll talk about the medium, callus plus auxin plus no cytokinin, little growth of callus occur. Callus plus auxin plus cytokinin, callus growth well and undifferentiated. A ratio of cytokinins and auxins are important in determining the fate of the callus. Callus plus low cytokinin and auxin, callus growth well forms roots. Callus plus high cytokinin auxins, callus grows well and forms meristematic shoots. Actions of cytokinins include it controls morphogenesis. Morph means form and genesis means formation. It regulates the cell cycle or cell division, okay, especially by controlling the transition from G2 to mitosis. This effect is moderated by cyclin dependent protein kinases, CDKs, and their subunit cyclins. Third, its action is bud development. It direct application of cytokinin promotes the growth of auxiliary buds. Exogenous cytokinins and auxins are thus antagonistic in their effect on auxiliary bud growth. Fourth, it delays senescence. Senescence is the programmed aging process that occurs in plants. Loss of chlorophyll, RNA, protein, and lipids. Cytokinin application to an intact leaf markedly reduces the extent and rate of chlorophyll and protein degradation and leaf drop. Fifth is greening. Cytokinins promote the light induced formation of chlorophyll and conversion of etioplast to chloroplast, greening process. Okay, cytokinin actions, it promotes cell expansion, stimulates the expansions of cotyledons. The mechanism is associated with increased plasticity of the cell wall, not associated with acidification. Okay, abscisic acid, it inhibits growth, promotes dormancy, and causes stomata produces in response to stress. Abscisic acid diagram is given in the Abscisic acids, their sites are plastids, tissues, especially leaves and seeds, transported through xylem and phloem. Abscisic acid action is drought resistant. Abscisic acid is the key internal signal that facilitates drought resistance in plant. It means it makes the plant stable to grow in the less availability of water. Under water stress condition, abscisic acid accumulates in leaves and causes tomato to close rapidly, reducing transpiration and preventing further water loss. Okay, abscisic acid causes the opening of efflux potassium channels in guard cells, plasma membrane, leading to a huge loss of this ion from the cytoplasm. The simultaneous osmotic loss of water leads to a decrease in guard cells turgor with consequent closure of stomata. Second function is freezing resistant. Makes a plant freezing resistant. Elevated ABA level are associated with increased freezing resistance. Mediates plant response to environmental stresses such as freezing by regulating gene expression. Certain genes are switched on by ABA while others are switched off. Third function is its in seed dormancy. A major role is played by it in seed dormancy. During seed maturation, abscisic acid levels increases dramatically. This inhibits germination and turns on the production of proteins and enable the embryo to survive dehydration during seed maturation. As dormancy can only be broken by specific environmental cues, it ensures that a seed will germinate only under suitable conditions of moisture, light, and temperature. The breaking of dormancy is associated with a decline in the level of abscisic acid. Okay. Next is ethylene, and the last one, ethylene structure is given. It is only gaseous plant hormone C2H4. naturally by higher plants and is able to diffuse readily intracellular spaces throughout the entire body of plant. Responses to environmental stresses, flooding in drought, infection, wounding, and mechanical pressure helps in shoot elongation, flowering, seed germination, fruit ripening, and leaf abscission and senescence. Ethylene action first is signal transduction. It helps in transduction of signals. Transmembrane proteins have been identified that bind to ethylene and the cell surface and function as signal transductions. Second is fruit ripening. It helps in ripening of many fruits. A series of changes include changes in color, decline in organic acid content, and increase in sugar content. Okay. Period of increased respiration, dramatic increase in ethylene production by climacteric. Ethylene can initiate the climacteric in a number of fruits and is used commercially to ripen tomatoes, avocados, melons, kiwi fruit, and bananas. Okay. Third is it acts on shoot growth and improves it, reduces elongation of the stem, bending of the stem, and swelling of the epicotyle or hypocotyle. It induces flowering. The ability of ethylene to affect flowering in pineapple has important commercial application. Promote flower senescence, aging in plants such as petunias, carnations, and peas. Induces flowering. Action is given. It act as thigmomorphogenesis, the change in growth form in response to a mechanical stimulation such as touch. Okay, brasinosteroids are plant steroid hormones that have similar structure to animal steroid hormones. Okay, promotion of cell elongation, cell division, and xylem differentiation, and delaying of leaf abscission. Brasinosteroids are a deficient mutant exhibit dramatic growth defects including dwarfism, reduced apical dominance, and male fertility, as well as delayed senescence and flowering. Brasinosteroids switch on specific genes by inactivating a protein that otherwise indirectly blocks transcription of those genes. Brasinosteroid structure is given in the next diagram. Hope you enjoyed the lecture. Stay motivated. God bless you. Allah Hafiz.